Hello, welcome to Science Breakthroughs from Science Mission. I am Sadashiv Pai and going to discuss a couple of breakthrough papers published in the week of August 4th. Let's start. First, we'll discuss a paper published by Brian and colleagues in Nature Communications on the role of enterocyte glutamine transporter on the development of metabolic syndrome. Why is this paper so important? Role of hyperuricemia in the development of metabolic syndrome is not well understood. Authors demonstrate deletion of urate transporter glutamine in mice impaired enterocyte uric acid transport kinetics resulted in full-blown development of metabolic syndrome. Allopurinol, a xanthine oxidase inhibitor, reversed the hypertension and hypercholesterolemia. Increase in urea thus indeed resulted in metabolic syndrome, and allopurinol partially reversed the effect. Let's look at the data. In the knockout mice, immunoblotting confirmed the deficiency of total glutamine and glutamine protein in enterocytes compared to other tissues. Isolated enterocytes from knockout mice exhibited a 65% urate uptake defect versus wild-type enterocytes. LCMS of stool extract revealed significantly decreased stool urate concentration in knockout mice. Together, these results suggested impaired enterocyte urate uptake and efflux into stool in knockout. Authors saw higher uric acid concentration both in serum and urine in knockout mice. Body composition analysis by echo-MRI revealed significantly elevated fat mass and fat percentage in knockout mice. Plasma analysis revealed knockout mice had significantly elevated total cholesterol, triglycerides, and free fatty acids. About 40% elevation in fasting plasma insulin in knockout mice in context of normal fasting blood sugar. Significant baseline hypertension in knockout mice was also seen when compared to wild type. Together, these results showed a total metabolic syndrome in knockout mice. Now, wild type and knockout mice were exposed to 8 weeks of allopurinol in drinking water approximately 4 weeks after weaning. Plasma uric acid was significantly decreased in knockout mice following allopurinol treatment, without significant effect on urine urate concentrations. In comparison with untreated knockout mice, allopurinol treatment also lowered blood pressure and total cholesterol without significant effect on free fatty acids and triglycerides, suggesting partial allopurinol mediated reversibility of spontaneous metabolic syndrome in knockout mice. Authors looked into secondary complications of hyperuricemia. Consistent with hyperuricemia patients, hepatic free fatty acids and triglycerides were elevated in knockout livers versus wild-type livers. In addition, mRNA for sterol regulatory element binding protein, a marker of cholesterol metabolism, was significantly elevated in the livers of knockout mice. Increased basal rate, decreased diastolic left ventricular internal diameter, and increased relative wall thickness, a common finding in patients with hyperuricemia, was also observed in knockout mice. Allopurinol treatment significantly lowered body weight and hepatic triglyceride content, but did not significantly reverse the knockout associated changes in heart rate, ventricular internal diameter, or relative wall thickness. Since not all the symptoms are reversed by allopurinol, there might be other mechanism in play in metabolic syndrome. Nevertheless, enterocyte glutamine transporter plays a crucial role in metabolic syndrome as shown in this study. Next, let's look into a paper published by Reinhard Kopp in Cell Reports. Authors unravel the function of breast cancer protein BRCA1. Why is this paper so important? BRCA1 forms heterodimer with BRCA-associated ring domain 1 protein and promotes repair of double-stranded DNA breaks. Loss of BRCA1 function in cells results in hypersensitivity to DNA damage associated with the development of cancer. Authors demonstrate BRCA1 BRAD1 dependent ubiquitination of histones at specific sites. Identification of these sites may help in the development of new tools for cancer diagnosis. Let's look at the data. Authors examined BRCA1 BRAD1 E3 ligase activity on individual histone proteins and reconstituted nucleosome substrates in vitro using electrophoretic mobility shift assay. As shown in this figure, BRCA1 BRAD1 ubiquitinating individual H2A, H2B, H3 and H4 proteins in vitro without any apparent preference. However, when BRCA1 BRAD1 was incubated with recombinant nucleosomes or active chromatin, only H2A was ubiquitinated, indicating specificity for H2A. To establish E3 ligase activity in vivo, authors fused ring domain of BRCA1 with M-cherry and transfected the cells and were able to co-localize it with antibody for ubiquitin. And the co-localization was not seen in the presence of mutated BRCA1. Quantitization of this experiment was shown here. Similar co-localization was also observed when H2A antibody was used instead of FK2. To identify the residue on H2A ubiquitinated, authors used mutated H2A nucleosomes and performed mobility shift assays with BRCA1. As shown here, BRCA1 dependent ubiquitination was markedly reduced with the nucleosomes in which both lysine 124 and 127 of H2A were mutated. And this shift was further reduced in 124, 127, 129 mutant. In vivo co-transfection of H2A mutant and immunoprecipitation studies with antibodies also revealed similar results. Also, co-transfection experiment with BRCA1 BRAD1 ring domains and H2A mutants and immunoprecipitation experiments also revealed the role of lysine 124, 127, 129 in ubiquitination. Finally, mass spectrophotometric analysis of chicken erythrocyte chromatin shows ubiquitination of C-terminal peptide Ka residues 126 to 128. Lysine 126 of chicken H2A is equivalent to lysine 127 in xenopus and human, further confirming the role of lysine 127 in ubiquitination. So this study not only confirms the role of BRCA1 as E3 ligase, but also identifies amino acids in H2A involved in ubiquitination. And this may help in generating antibodies directed against H2A 127 to 129 for use as a diagnostic tool to identify biochemically active BRCA1. Thank you for watching Science Breakthroughs from Science Mission.